Okay. Welcome everybody to this second online immunometabolism seminar. No, we were organizing a physical ImmunoMetNet symposium in Amsterdam, which we had to cancel. And one of the speakers we also had to cancel is David Finlay. David Finlay is associate professor at Trinity College in Dublin. And his group tries to shape immune cell function by manipulating their metabolism. And to do so, he's supported by quite some prestigious grants, including an ERC consolidator. David is aiming for a 40-minute presentation. Afterwards, we'll have time for questions. Please ask your questions via the chat box, and we'll follow up on this after his presentation. So welcome, David. Thanks again for accepting to present here. And you can start sharing your screen, and we look forward to your presentation. Go ahead.
Okay. Okay, guys. Thanks very much. You know, everything's changed in the last few weeks about how we operate. But you know, I think everybody seems to be jumping and grasping the opportunity to share their science on a real more global platform. So it's great to be able to speak to everybody where you are in your pajamas and whatever whatever state of comfort you're in. So I'm delighted to be able to have a chance to share our science with you today. So today I'm going to talk about nutrients and metabolites and how we think they are playing a role in controlling lymphocyte responses. And this is really broken down into two parts. So first of all, we start thinking about some of the complex microenvironments that can be occurring in sites of inflammation at various sites. Within the lymph nodes, for instance, you have a real diverse sets of immune cells that are making close contact and sometimes clustering around each other. And we know from studying these all individually that that these all dramatically regulate their metabolism when they become activated. So you can imagine in a site like an inflammatory lymph node, you've got all these cells interacting with each other, all with a high demand for nutrients. So the question becomes whether there is sufficient nutrients for all the cells' demands, or whether cells like dendritic cells that often are found at in the center of these clusters surrounded by lots of T cells, NK cells, other immune cells that have also great demand for nutrients, whether nutrients become limiting. So is there competition for nutrients between immune cells? So that's one part of what I'm going to talk to you about. So whether nutrient restriction can happen, and if it does, how does that affect immune responses? The second part that I want to talk about is about metabolites. So can metabolites also affect other cells in the local microenvironment? So this is there's some good examples of this, but in particular, we've been working on this in the context of natural killer cells, and I'll talk to you about that. So metabolites that are generated through metabolism of local cells acting on cells in the near or local environment. So I think what we know is nutrients and nutrient levels and active metabolites, they vary greatly depending on the microenvironment you're in. So you think of the example of within the blood, you've obviously, very high concentrations of nutrients. Glucose goes up and down during meals. So if you're a cell in the blood, you're never going to be deprived of glucose. In fact, glucose is very well maintained in most tissues, except maybe in discrete microenvironments, like, for instance, in the tumor microenvironment. That's one microenvironment we know for sure that glucose levels are low. We also know, although it's quite hard to measure glucose in discrete microenvironments, that it becomes difficult to be sure. But we've good reason to believe that glucose levels could also become limiting at sites of infection and sites of autoimmunity. So in infection, you can imagine Staph aureus is a bug that uses glucose as its primary fuel. You can imagine a huge infection of Staph aureus. There's not going to be much glucose left over. So we're starting to appreciate that nutrients may not always be available to all cells at all times, depending on these discrete microenvironments. So in order to sort of fully appreciate this talk, I'm going to give you sort of my Metabolism 101 slide, and for all of you who have already dived into immunometabolism, this will all be fairly easy for you. But maybe there's a few people who are new to this. So this is my my slide describing why metabolism is important for immune cells, from some of the obvious examples to some of the less obvious examples. So to keep it very simple, we're not adding in too many of the enzymes, the complicated steps, but obviously we all know that glucose is a key fuel. It's a very important fuel for lots of immune cells. Not the most important one, but often when we hear of activated immune cells, we hear about them engaging aerobic glycolysis or the Warburg effect, and that's, you know, metabolizing glucose in vast quantities to lactate. And this is called aerobic glycolysis. And this is one of two routes that glycolysis can go. The other route is into the TCA cycle. Now, these are both very different routes. You know, the flux to lactate can be very high. The flux through the TCA cycle and oxphos can't be as high. But they both ultimately, you know, they generate energy. You know, glycolysis generates, you know, energy inefficiently but fast. Oxphos produces energy very efficiently but slower. So, you know, they're both be a very useful source of energy. So that's really the first reason why metabolism is important for immune cells, to provide the energy they need for their functions. And bear in mind that those energy demands change greatly upon activation. If you think about a naive T cell, which does very little, really, going towards an activated T cell, which is trying to proliferate and produce huge amounts of lots of migration, all these energy demanding processes. As I said, glucose is not the only fuel. Other fuels that are important are amino acids. You know, fatty acids and glutamine can all feed into the TCA cycle. Amino acids can also feed into glycolysis. So these are other fuels. But another key function for metabolism, especially in activated immune cells, which have high demands for generating biomass, is biosynthesis. So, you know, glucose can be used to make intermediates that can be used for making proteins, nucleotides, and lipids. Also, TCA cycle provides the building blocks for these processes as well. So biosynthesis is also important. What else? And I guess, so they're kind of some of the basic biochemical reasons, which we all would have known anyway before this immunometabolism field ever got started. But some of the more exciting sort of discoveries in the last 10 years have been that certain metabolites and certain metabolic enzymes actually can play a more direct role in controlling immune function. So, you know, if you were tuning into Luke O'Neill last week, you would have heard about succinate and probably citrate in the TCA cycle that are really important for generating immunological molecules that can be both stimulatory and suppressive in macrophages. Also, you know, phosphoenolpyruvate is known to regulate TCR signaling. Some of the metabolic enzymes are also important. GAPDH is one of the first enzymes to be shown to have a moonlighting role, and it can control IL-2 translation and interferon gamma translation. Hexokinase is linked to inflammasome activation. So really, there's quite a lot of really direct links between metabolism and a regulation of the immune system, which has made the field really quite exciting in the last number of years. So regulation. So and that can be regulation from translation to epigenetics to generating various pro-inflammatory and anti-inflammatory molecules. Other processes that are also controlled, Joe, glucose is also an important source of intermediates that feed into the pentose phosphate pathway that make NADPH, which is important in keeping redox balance by generating glutathione and also thioredoxin, or keeping those molecules reduced to allow them to be antioxidants. So redox balance is another important function. And also what I'm going to talk to you a little about today towards the end of this talk is intracellular regulation. So the idea that metabolites are generated from these core metabolic pathways and other metabolic pathways can be used to communicate with other cells in the surrounding microenvironment. So, as I said, they're the two parts of this talk that are going to come together hopefully towards the end. And I guess to give you an example that we don't necessarily work on, but an example which you probably are familiar with, where both of these systems are working at the same time, is the sort of the tolerogenic DC that expresses, or dendritic cell that expresses indoleamine 2,3-dioxygenase, or IDO. So this is an example where you both get depletion of competition for nutrients and the production of active metabolites that can control T cell responses. So IDO metabolizes tryptophan, depleting levels of tryptophan, and also generates tryptophan metabolites like kynurenine and also further metabolites of kynurenine. These can inhibit T cell responses, whereas the lack of tryptophan also inhibits T cell responses. So depletion. So here we have in one system depletion of tryptophan and inhibition of tryptophan due to tryptophan metabolites. So here's just an example of the kind of concepts we're talking about here. Okay, so first of all, I'm going to talk to you. The first story is about dendritic cells and how they control T cell responses. So for those of you who aren't so familiar with dendritic cells, here's a sort of brief introduction. Dendritic cells are key orchestrators of the immune response. You know, they sit in tissues and they sort of sample the environment. I like to think of them as sponges. They're soaking up everything, looking for evidence of danger, you know, looking for damps and pamps and evidence of pathogens of infection or danger. And they are capturing antigens by phagocytosis, capturing dying cells, microbes. And when they become activated, they then migrate and mature as they go to the lymph nodes. And in the lymph nodes, they have a very different function. They are now not acting as a sponge, but more like acting as a source of information. They're instructing and stimulating T cell responses. They're also interacting with other immune cells to drive particular immune responses. And we've particularly been looking at conventional DCs, so these dendritic conventional DCs as opposed to plasmacytoid DCs that are important in this process of stimulating T cell responses. And as my background has been with CD8 T cell responses, we're interested in the subset that specifically does that. So here's a real sort of nice simple diagram to tell you what that key instruction is taking place in the lymph node, what are the key signals that dendritic cells are delivering. So it's very simple. It's a three sort of signal process. Obviously, the dendritic cell expresses or has antigen, which is loaded with specific antigens. And they pick out from the pool of T cells the ones that have the right TCR. So this is where the specificity of the system works. You also have signal 2, which is the co-stimulatory molecules. That's to ensure that these T cells are activated in the right at the right time as of a key second signal. And the third signal, the cytokines, is what kind of shapes the nature of that T cell response. Because these cytokines can dictate what type of T cell response you get. So if this is a CD4 T cell, depending on what cytokines are in the environment, you might get a regulatory T cell, you might get a Th17 or a Th1 response. So the cytokines are also an important signal 3 that shapes the T cell response. So I guess one of the first questions we thought of, and this is going back a number of years now, was how do nutrients impact upon the system? How do nutrients impact upon dendritic cell-induced T cell responses? And of course, at that stage, we already knew that glucose and glutamine are really important for T cell responses. If you deprive T cells as they're activating of these nutrients, they really aren't very happy at all. But we knew a lot less about dendritic cells. And some of our early work showed that when you deprive a dendritic cell of glucose, you can actually get quite an interesting response, because actually these dendritic cells seem to have increased function. So at the time, most of the literature that was coming out was saying that glucose was essential for immune function. If you took away glucose, immune cells didn't work as they were supposed to. Whereas dendritic cells seem to buck the trend in that when we limited glucose availability to the dendritic cells, they had increased ability to activate T cells. So this was work by my first PhD student, Simon Lawless, and what he did, he restricted glucose availability to these bone marrow-derived DCs. What he found was that he had an increase in signal 1 and 2. So co-stimulatory molecules were increased, the expression of IL-12 was increased, and then when he added T cells to these dendritic cells and looked at the T cell response, he found that the dendritic cells that had been glucose deprived were able to induce an increased T cell response. It's important to say here, we activated dendritic cells in the absence of glucose and then put them with T cells with lots of glucose. So they weren't actually glucose deprived when they're seeing the T cells, but nevertheless, the process of being glucose deprived initially has allowed them to have increased signal 1 and 2, and these T cells proliferated more. And when you look at the outputs of these T cells, they made more interferon gamma. So this kind of suggests to us that, by restricting glucose availability to the dendritic cells, they had increased ability to drive CD8 T cell responses. Now, of course, one caveat with all this work was that it was done in, GM-CSF-derived bone marrow-derived DCs, and while these are a good model for some things, and it's good for doing biochemistry, there's a lot of caveats with it. So I think it was really important at this stage to see whether we could see similar effects if we took real dendritic cells straight out of a mouse. So we're talking about now classical DCs from the purified from the spleen. So this all sounds very nice and simple, but as I'll show you in a second, it's not quite as simple to work with dendritic cells straight out of a mouse as you might think. But so just a brief introduction to what these cells are. So classical DCs from taken from a mouse, they are sort of broadly categorized into two categories. Your CDC2s, and this is how you distinguish them, and CDC1s are distinguished by the fact that they have CXCR1 positive, and they kind of, they broadly sort of have a role in driving the CD4 responses and CD8 responses. These CDC1s, are involved in cross-presenting to CD8, so they're important for that. They both make cytokines. But we are interested initially in the CDC1s because we, as I said, had a history of working with CD8 T cells. So Diana in my lab was a great postdoc who started a number of years ago, and she had the task of trying to get these classical DCs out of a mouse in sufficient numbers to be able to do the experiments we wanted to do. And it took a lot of optimizing and a lot of hard work. So this one slide really epitomizes a lot of blood, sweat, and tears. But essentially the approach we took was there are very, very few dendritic cells in a spleen of a mouse. So really just taking the spleens is not a viable option. First, what we had to do 10 days beforehand was to inject tumor cells that express FLT3 ligand. Now, FLT3 ligand is the cytokine that drives the differentiation of DCs. And when you do this, you get a nice expansion of DCs in the spleens of these mice. So here you can see in a control mice, less than 1%. In these mice that had this tumor in the flank for 10 days, you've got a nice expansion up to about 12%. And then Deanna found various ways of purifying them, including a sort of gradient purification followed by an XCR1 positive magnetic bead separation. Allows you to get pretty pure dendritic cells. We can get anything between 90 and 95% purity, which is great. And so then what she did was basically try and replicate some of these initial experiments in bone marrow-derived DCs. She took these dendritic cells, she activated them with CpG, which is a TLR agonist, plus or minus glucose, for 6 or 8 hours. She also pulsed them with the OT1 antigen Seinfeld. This is a peptide from ova. And then after these 6 hours, she washed away the medium, put them all into nice nutrient replete media, plus some OT1 T cells, and looked at how these dendritic cells were able to drive T cell responses. So these first number of slides, we're looking at the T cells and the overall outcome, and then we'll come back to looking at how the starvation affected the DCs. So we did this both for glucose or glutamine. The results for the glucose were a little bit more interesting, but nevertheless, glutamine was interesting seeing that both these sort of nutrient restriction regimes had an effect on the cells. So here's a slide just to show you that when you do this, so the top is the no antigen, the bottom is antigen, and both stimulated with CpG. You can see that over time you get an increase in this activation marker of T cells, CD69, and you get also an increase in blastogenesis, so as you can see here. So over time the cells become big, which is what you'd expect from an activating T cell. However, if you deprive the dendritic cells of glucose for the first 8 hours before they ever saw the T cell, you can see they activate normally. So, and same for in the absence of glutamine. So if you look at 69, there's no difference in 69 expression. The T cells are just as active. So in sort of overt terms, these dendritic cells have been starved for 6 to 8 hours are perfectly capable of activating the T cells. But next we wanted to know, well, what about the nature of that T cell response? So is that T cell response different in any ways? So we started to look at some cytokines. So here's some of the cytokines that an activated T cell might make. TNF alpha, as you can see in the normal media, you see that they're making some TNF alpha. If you take away glutamine or glucose, you can see that they are actually making more TNF alpha. So it's certainly a clear increase, so it's suggesting that they are indeed increasing activation. We also looked at interferon gamma, which is obviously one of the best known excuse me, effector cytokines for T cells. And again, we saw quite a pronounced increase in interferon gamma production by these T cells when they had been co-cultured with these dendritic cells that had been starved of glucose for those first 6 hours. So this is very interesting. It shows us that in general, in principle, this is all working in a similar way. You know, if we nutrient restrict these dendritic cells, they seem to be better able to activate T cell responses. So we then went back to the dendritic cell and asked what was it about the dendritic cell that was allowing them to be better at driving T cell responses. And the two obvious candidates were, of course, co-stimulatory molecules and cytokines, and we looked at both of these. And first of all, with the co-stimulatory molecules, you can see here a nice increase with CpG stimulation of the CD6, CD80, for instance. But it doesn't really it certainly doesn't go up with any of these regimes here, where you think when you deprive them of glucose or glutamine. If anything, goes down a bit. So certainly, increased expression of co-stimulatory molecules was not explaining this. So that was a bit disappointing at the time, but we moved on to looking at some of the cytokines. So here's a couple of the cytokines that dendritic cells make when you stimulate them with CpG: IL-6, TNF, IL-12. And when we stimulate them with CpG in the absence of glucose or glutamine, there's actually some quite striking results. And as you can see here, we get quite a pronounced increase in all cytokines when you take away glucose. In particular, which was really striking, was the increase in IL-12. Of course, IL-12 is a very important cytokine for driving CD8 T cell responses. So that was very interesting. So we think, you know, it seems to be the cytokine production that is helping these dendritic cells to have enhanced ability to induce T cell responses. So that was the kind of idea. Now I haven't got the data here, but Deanna has gone further to ask, well, you know, can these dendritic cells also have an increased capacity to stimulate T cell responses in vivo, and she's adoptively transferred these, first of all, OT1 cells and these dendritic cells at a 1-to-1 ratio into mice, allowing them to meet in the lymph node. And similarly, we're starting to get some nice preliminary data saying that they can do so. So it seems like these glucose-starved dendritic cells have an increased potential to drive T cell responses. So, you know, one of the other questions, you know, which is probably a harder question to get to, is, so when might dendritic cells become deprived of glucose? So I've mentioned within tumors, within lymph nodes, maybe in these inflammatory sites. So it's hard to prove this, but and we're working on developing tools to look at the nutrient levels in these environments. But one idea we had was you know, from a lot of the lovely imaging work that's been done by various labs, is that dendritic cells can form clusters within lymph nodes as they are activating T cells, where there are multiple T cells interacting with a single dendritic cell. So we wondered whether in this sort of scenario, would you have a condition where a dendritic cell that's interacting with just 1 or 2 T cells would have plenty of glucose, but in these clusters where there's lots of T cells interacting with them, might there be a limiting of glucose towards the dendritic cell, simply because the T cell is which we know has a sort of prodigious appetite for glucose, is sucking up all the glucose. Of course, this is simpler than it really is, because if you look at these the data out there on what interacts with dendritic cells in the lymph node, not only do you have T cells, you'd also have lots of NK cells are known to make prolonged interactions with with dendritic cells, as are other dendritic cell populations. But we thought this was a nice sort of a reductionist approach to take. And one prediction we thought we'd get is if you did have a glucose or amino acid starved dendritic cell, this key nutrient sensor mTOR would be turned off. So mTOR is sensitive to both glucose levels, to amino acid levels. So we think of it as quite a sort of a sensitive nutrient sensor. So in this scenario here, we'd expect to have mTOR turned on. In this scenario here, we expect to have mTOR turned off. So, so this is the idea, is that the clustering around dendritic cells may create a scenario where glucose becomes limited to the dendritic cell. And we decided to do this just by doing co-cultures of dendritic cells and OT1 T cells. And this is some imaging done by in collaboration with James Murray in our institute, and Ryan McGargle, who was one of his PhD students at the time. And here's one of the nice images that we had when we co-cultured these T cells and dendritic cells. And you can see here's a dendritic cell that's interacting with just 1 or 2 T cells. Here's a dendritic cell that's it's in purple, or it may be hard to see, but the dendritic cell is stained in purple, but it's interacting with lots of T cells. As you can see here, up to 10 T cells interacting with this dendritic cell. And in red is the measure of mTOR signaling, which is a phospho-S6 staining. And you can see, there's lots of phospho-6 staining in these dendritic cells, but none in this dendritic cell. And we're able to quantify this for different ratios of T cells. So you can see when you have 2 T cells for every dendritic cell, you have quite a range of mTOR activity. But it was very clear when you increase the numbers of T cells that these dendritic cells didn't have mTOR anymore, and it was comparable to the situation is if you added rapamycin to artificially inhibit mTOR. And importantly, this effect was only seen in dendritic cells and not in the T cells. In the T cells, there's equivalent amounts of mTOR activity based on phospho-6 staining, irrespective of what the ratios were. So we think that that's work that's all ongoing, but the idea is that in these immunological microenvironments where nutrients are at a premium, that the nutrients can become limiting. And in the case of dendritic cells, we're starting to get the impression that this is a way of increasing the ability of these dendritic cells to stimulate T cells. And what the exact physiological consequences of that are still to remain to be determined. We've lots of work to do there, but that's essentially what the nature of our ERC grant is to further study that concept. So the second part of my talk is to talk about how metabolites can transmit communication between cells, and in particular focusing on some of our work that's been going on in the NK cell. So natural killer cells are a very brief introduction. These are innate lymphocytes. They are best known for their ability to recognize virally infected and transformed cells. Some of their key outputs are they're a big producer of interferon gamma. They're cytotoxic cells, so they can recognize target cells, form synapses on them, and directly kill them using this cytotoxic process involving cytotoxic granules, as you can see here. And here's also a tumor cell, which is labeled with a dye that fluoresces when the cell dies. You can see this NK cell killing that tumor cell here. So these are cells of the innate immune system, which means they are primed for immediate action, and they can kill straight away. Unlike T cells, they can kill straight away. They can make interferon gamma very rapidly, but they're not strictly just innate cells, because they also have prolonged function. I think which is something that's emerging for a lot of innate cells that can work alongside the adaptive immunity. So they can respond to innate cytokines like 12, 18, and 15, and over time that gives them an increased capacity to make even more interferon gamma, and they become even better at killing. And also they can respond to adaptive cytokines like IL-2, which allows them to work over a lot longer periods and maintain these elevated effective functions like killing target cells and making cytokines. And all these elevated functions we've shown to be linked to increased metabolism. So there's a really distinct increased metabolism that occurs as you move from this to this here, that involves a real ramping up of glycolysis and oxphos. So essentially that's sort of the first question we asked was metabolism important in NK cells? And we've been studying this for some time now, and we know that that glucose metabolism is really crucial. Glucose is really the main fuel for NK cells, and we've also done a lot of work on what are some of the key metabolic regulators. And today I just want to talk to you about one of those, and that's Streb, because that's the one that's particularly relevant to this idea of intracellular communication that I want to talk to you about. And so someone may seem like a stranger amongst this bunch, because it has never really been linked to glucose metabolism before. But in NK cells, it really is one of the key regulators allowing these cells to metabolize glucose. So I'll have to give you an introduction to Streb. So Streb is best known because it's sort of the master regulator of lipid synthesis. So it pretty much drives the expression of every gene that's involved in converting acetyl-CoA to cholesterol and also fatty acids. So it's really important in lipid synthesis, and there's loads of literature on that. And so because it's so important at that, and there's been Nobel Prizes won on this by Goldstein and Brown, the mechanism of activation is well characterized, but very complicated. So Streb is an integral membrane protein in the ER. It needs to translocate to the Golgi, where it meets two proteases, S1P and S2P. It becomes cleaved and then enters the nucleus, and that's the active transcription factor that can bind to these sterile regulatory elements. So in order for Streb to move to the Golgi, it needs this chaperone protein, SCAP. SCAP brings it to the Golgi, so that's very important. And it's the regulation of this processing that controls activity of Streb. So for instance, if you have plenty of cholesterol, this is a feedback loop. Cholesterol will stop the trafficking of Streb to the Golgi, turn off Streb, turn off de novo cholesterol synthesis. So nice sort of feedback loop, as you might expect. Also, oxidized cholesterol species like 25-hydroxycholesterol will also act slightly differently. It causes this interaction between INSYG and SCAP and also prevents the translocation. So both these inhibit Streb activation and shut off fatty acid synthesis and cholesterol synthesis. So we are wondering whether cells of the immune cells and tumor cells might be able to make these oxy-sterols in particular to communicate and regulate NK cell function. So first of all, I said it was kind of unexpected that Streb was a controller of glucose metabolism. So I want to convince you that this is indeed true. So I'll show you some of our basic data. So this is the slide I showed you at the beginning, showing you in a very crude terms the basic of glucose metabolism involving aerobic glycolysis and the TCA cycle. You can measure this with the Seahorse machine. You may all have one of these, and you know how it works. It's very simple in some ways, but also, it gives you a lot of information just through the way it works. So it just measures pH, essentially the acidification, which is a measure of aerobic glycolysis, and oxygen consumption. But with all the different types of inhibitors you can use, you can really pinpoint where this oxygen has been used. So it gives you quite a good picture. So we looked at the role of Streb in driving glucose metabolism by looking at two inhibitors. So this 25-hydroxycholesterol, which I talked to already, this prevents processing of Streb by preventing it moving to the Golgi. And also this pharmacological inhibitor, which actually it's a protease inhibitor. It stops S1P and also stops processing. So both of them inhibit Streb very clearly. And here you can see an unstimulated NK cell and a stimulated NK cell. So a real robust induction in both glycolysis, as measured by here, ECAR, and OXPHOS measured by OCAR, but very clearly that if you inhibit Streb, you really have a robust inhibition. So you really prevent these guys from upregulating both glycolysis and OXPHOS. And we confirmed that with a genetic model as well, where we knocked out SCAP and it also prevents Streb activation. And why is this? Well, the reason for this is actually when we did the sort of detailed metabolomics, which I won't go into now, we found that glucose wasn't being metabolized by the TCA cycle in our cells, but rather by this other sort of more complicated, well, slightly different cycle called the citric-malate shuttle. And the reason Streb is important for the citric-malate shuttle was because it controls two of these key components of the citric-malate shuttle: this transport here and this enzyme here, ATP citric lyase. And this is all work done by a postdoc in my lab who's now moved on to Christoph Hess's lab, Nadine Assman. So here's probably the most complicated metabolic slide I'm going to show you, which is the citric-malate shuttle. So here's your normal glycolysis to make pyruvate, which is then converted into acetyl-CoA in the mitochondria. This is then is combined with oxaloacetate to give you citrate. But in normal TCA cycle, this is further metabolized around the TCA cycle, but in the citric-malate shuttle, it is exported. It's exported out of this transporter SLC25A1. It's then metabolized in the cytosol by ATP citric lyase to give you oxaloacetate and then malate. And then malate returns back into the mitochondria, rejoins what would be in the TCA cycle here. And you have this quite convoluted cycle that crosses the mitochondrial membranes. Now, how does this compare to the TCA cycle? Well, it also generates NADH, so that can enter into the TCA cycle, generate your gradient for driving ATP synthesis. So it's also very good at driving ATP synthesis. But it has an added advantage of regenerating NAD+, which, if you know your glycolysis, you'll know that that's a really important cofactor for GAPDH. So it sustains both glycolysis, OXPHOS, but it's different from the TCA cycle because it also generates cytosolic acetyl-CoA, which may be useful for acetylation, so epigenetic control, for instance, or lipid biosynthesis. So as I said, Streb is important because it controls these two factors, and here's the data showing that. So if you inhibit Streb, these NK cells can't upregulate this transporter, and they can't upregulate ATP citric lyase. So that's all the metabolism of it. But when we looked at the function, one of the most striking functional defects in these NK cells was that they couldn't kill tumor cells. So as you can see here in the 25-hydroxycholesterol in the red, here's a normal cytotoxic killing assay. This is where you take your activated NK cells, you put them in co-culture with tumor cells that have been loaded with a fluorescent dye. If the NK cell kills the tumor cells, the fluorescent dyes are released and you get a very specific measure of killing. Here is your activated NK cells, very good at killing at 5:1 ratio. It's killing most of them in these 4 hours. But if you activate them in the presence of 25-hydroxycholesterol, they can't kill at all. It's really quite striking. So the first question was, so 25-hydroxycholesterol is best known as a regulator of Streb, but we want to know what about other oxysterols. So this is where I have to sort of introduce you to what oxysterols are. So these are really, they're oxidized versions of cholesterol. So this can happen spontaneously, just spontaneous oxidation. So if you have a cholesterol-rich environment, you do get oxysterols just through the spontaneous process. But the most elevated oxysterols in the body are 25-hydroxycholesterol and 27-hydroxycholesterol. Sorry, I should say most elevated outside of the brain, because 24-hydroxycholesterol is really high levels in the brain. But outside the brain, these are the highest levels. And that's primarily because there's specific enzymes for generating these. Cholesterol 25-hydroxylase generates 25-hydroxycholesterol, and CYP27A1 generates 27-hydroxycholesterol. So these are really the highest levels, and they can really be elevated in certain situations, even in the serum, even within the blood, systemically. So what do we know about these two enzymes and these two oxysterols? Well, quite a bit's known about 25-hydroxycholesterol. It's an inhibitor of Streb. It also activates this nuclear receptor LXR, and there's lots known about this in the context of inflammation. So it functions in macrophages. It has antiviral properties. It's involved in regulating IL-1 beta and antibodies. For 27-hydroxycholesterol, there's less done on it. And the real reason for this really was that 25-hydroxycholesterol was the first oxysterol to be made available commercially. So for years, people were churning out data on 25-hydroxycholesterol, whereas others fell behind. But in fact, there's about 3 or 4 other oxysterols that are also potent inhibitors of Streb. And 27-hydroxycholesterol, we know inhibits LXR. There's very little evidence it inhibits Streb, except one paper in liver cells last year that suggested it does. But it's well known in the context of cancer because it's a specific ER mimetic. You can bind to the ER receptor and drive signaling. So it's implicated in breast cancer and cervical cancer. But less is known about it in the immune system, to be honest. So Chloe in my lab, it was her job to find out whether 27-hydroxycholesterol could inhibit Streb in NK cells. And by looking at Streb target genes, the data was very clear it could. And it's also a potent inhibitor of NK cell metabolism to the same degree as 25-hydroxycholesterol. So it seems like both these oxysterols are really potent repressors of NK cell metabolism. Here we can see basal glycolysis. This is your typical seahorse plot showing basal glycolysis and your maximal glycolysis that is available to it. Same for the OXPHOS, you can see potently inhibited in both cases. So, Streb really unexpectedly is an essential transcription factor for driving glucose metabolism, and these two oxysterols inhibit Streb and really have a real detrimental effect on the metabolism of these cells and on their ability to kill tumor cells. Actually, there's multiple aspects of their functions are down, but the tumor killing is really the most pronounced. And here you can see in unstimulated NK cells, as I said, they're able to kill anyway because they are innate killer cells. When you stimulate them with cytokine, they have an increased capacity to kill. But when you add these oxysterols, you really deplete their ability, even below unstimulated. So these cells really effectively can't kill target cells. And we've shown this for multiple different tumor cell lines. Here's a cell line where there's 2 versions of it. One that's relatively resistant to killing by NK cells, one that's much more sensitive because it doesn't have MHC1, and NK cells recognize the absence of MHC1, and that's a trigger to kill. But in all these situations, oxysterols prevent these cells from killing target cells. So that leaves with a bit of a puzzle because cytotoxicity is quite a complicated process. So Chloe's job now is to really dissect at what point this is happening. So if you think about, first of all, the NK cell needs to be able to migrate and find the tumor cell and form these conjugates. We need to determine whether that's happening normally in the presence of oxysterols. They need to form the synapse where these receptors find their respective ligands. Once this is formed and a close synapse is formed, you get this mTOC polarization where it moves towards the tumor, and then you get degranulation where these granules containing the cytotoxic molecules are released into the synapse. Now, we collaborated with Lydia Lynch on a project looking at obese NK cells, and they couldn't kill either for different reasons. It wasn't due to, that was due to activation of lipid loading and activation of PPARs. And one of the key aspects to their inability to kill was that they couldn't polarize the mTOC. So they could form these synapses, but they couldn't bring the granules to the synapse, so they couldn't kill the target cells. So Chloe's going to start, and that's something that's ongoing. So we don't really know what the answer there is. But we did try to rule out a few things. So if you think about oxysterols, so one of the questions would be is like 25-hydroxycholesterol can insert directly into the membrane. So are we just altering the membrane of the NK cells so that they can't? So it's altering the membrane fluidity in order. And you would think if you're forming synapses and degranulating membrane fluidity is going to be important. That's not the case, because we have this pharmacological inhibitor that's nothing to do with membrane fluidics. So this is a protease inhibitor, and it inhibits killing just as much. So we ruled that out. But then the next question was, well, maybe you are affecting membrane dynamics and membrane fluidity, but it's through Streb. So this is an inhibitor of Streb. So maybe you're changing cholesterol synthesis, you're changing fatty acid synthesis, and changing the nature of the membrane as well. So maybe it's just purely a physical thing of the membrane of the NK cell. We also know that's not true as well, because we can actually block both cholesterol synthesis and fatty acid synthesis by blocking the first enzyme in this pathway, acetyl-CoA carboxylase, with this inhibitor TOFA, and that has no effect on killing. So if you actually block the sort of normal, well-described functions of Streb, which is making lipid, the NK cells don't care at all, and they're perfectly able to kill the tumor cells just as efficiently. So it's something to do with Streb, and something to do with Streb that's not its normal functions. So Chloe's going to have a great fun dissecting that over the next couple of years of her PhD, which I'm sure she's a talented student. So I have no doubt she's going to come up with the money. But one thing I guess we can't exclude, and I think this is quite an interesting idea, is that it could be due to just pure energy. So and this is a really lovely electron micrograph that we pulled up from a paper that was 1982 of an NK cell, human NK cell, interacting with a target cell. This is a leukemia target cell, forming a nice close synapse. It's about to kill it. These are the granules here are moving towards it. But look at this. These are all mitochondria. They're all lining up on the synapse of the NK cell and the target cell here. If you look at everywhere else, there's no more mitochondria. So they have moved their mitochondria to the synapse, which really, I guess it makes sense. We know the mitochondria move to where energy is required. So this, fits with the idea that this whole degranulation and polarization of the mTOC to the synapse is an energy dependent process, and that's why the mitochondria are there. And maybe this is why when we inhibit Streb, we can't kill, because they haven't got the energy to engage in this energy demanding process. Now we need to look at all these things and mitochondrial health as well. So that's going to be an interesting project going forward. So coming back to this, so okay, that's fair enough. We know that they don't kill. So do we have any evidence that so all the experiments I've showed you so far, we physically put the 25-hydroxycholesterol into the well with the NK cells? Have we any evidence this can be made by a cell in the local microenvironment? So to address this, we need to know what cells make these oxysterols. Well, if you probe the ImGen database, the cholesterol 25-hydroxycholesterol data is really nice. Look at this. It's really specifically expressed in a very few cells. And when you look at these are inflammatory macrophages and inflammatory DCs, and a couple of lymph node endothelial cells. So really suggesting that within this inflammatory lymph node could be a scenario where this is happening. If you look at the expression of CYP27A1, it's a bit more widely expressed, but again, really just in myeloid cells. So we think that myeloid cells interacting with NK cells, this could be where this communication could be happening. So that was one question we had. So is this a physiological process that could be happening within an inflammatory lymph node where, for instance, an inflammatory macrophage or inflammatory DC could be communicating with NK cells? And also, could it be a pathophysiological occurrence where tumors, for instance, hijack the system to inactivate NK cells and prevent them from killing the tumor? So, and actually, if you start probing some of the RNA-seq databases of tumors and tumor-associated macrophages, there is clear evidence that some tumors overexpress these enzymes, including glioblastoma, breast cancers, and there's less information out there on TAMS. But some of the data sets we found does suggest that this is occurring in TAMS too. So the last few slides are just a few experiments we did where we co-cultured M1 macrophages with NK cells and asked whether that could turn off an NK cell. And this is work done by a PhD student who's just finished my lab, Katie O'Brien. So if you activate bone marrow-derived macrophages with LPS, They upregulate the enzyme cholesterol 25-hydroxylase, and here we can see that in our cells we activate them with LPS. You can see increased. This shows that they're activated because they're making IL-1, and here shows you to 25-hydroxylase. And so this was the two co-culture conditions we did: an unstimulated macrophage with an NK cell, and we add the cytokines that will activate the NK cells as well. A stimulated macrophage that's making 25-hydroxycholesterol in the presence of NK cells with the cytokines required to activate the NK cells. And what happens? So it's very clear that in this situation, the NK cells get big, they activate, and as you can see that here with the forward scatter and the expression of this nutrient transporter CD71. But if we have an activated M1 macrophage that's expressing cholesterol 25-hydroxylase, these NK cells are smaller, and they express less transferrin receptor, they make less interferon gamma, make less granzyme B. These are all the characteristics that you'd see of an NK cell if you added 25-hydroxycholesterol yourself to the well. So this certainly was consistent with 25-hydroxycholesterol mediated inhibition of NK cell function. But of course, this is a bit complicated because 25-hydroxycholesterol also has lots of things in the macrophage. So, we did think about what could be happening in the macrophage that could be inhibiting NK cell, and there wasn't any logical explanation. But still, it's a complicated experiment. So we then got some cholesterol 25-hydroxylase knockouts and did the same experiment with these macrophages that either were knockouts or wild type for cholesterol 25-hydroxylase. So we have 4 conditions here. As you can see, only one of these conditions has the ability to make 25-hydroxylase, and that's up here. And you can see when these cells, was the red line here, are able to cause the activated NK cells to be smaller. In the knockouts, they're slightly smaller, but it's not really that significant. This is all, actually I should say this is a very preliminary data, so we've only done this once or twice, and then the people who we're collaborating with they lost their line, so we're waiting to get some more of these mice. But you can also see here, in this condition here, where these cells making 25-hydroxycholesterol, the NK cells are making interferon gamma, but this is not the case in the knockouts. And same with granzyme B, less granzyme B, not the case in the knockouts. This is also a complicated experiment because if you knock out cholesterol 25-hydroxylase in macrophages, well, the macrophages are going to be slightly different. So we're currently working on the knockout experiment, which involves doing essentially this experiment again, except transducing the NK cell with a Streb that can't be inactivated. So we have the vectors for that. So if you put a Streb that's already processed, you should be able to maintain size and effective function even in the presence of these activated macrophages, if it is 25-hydroxycholesterol Streb mediated process. So that's the killer experiment that we're working on now. We're working up towards that. So I've yabbered on for quite a bit, but just to remind you, I've told you 2 stories here today about the concept that nutrients can become limiting in certain microenvironments, and that could be a very interesting way of controlling immune cells. And in certain contexts, for instance, T cells and NK cells, if they're deprived of glucose, they don't work so well. But there are other situations, like for instance, a glucose-deprived dendritic cell, where they do seem to work. They seem to work and have enhanced function. So that's a really interesting sort of dichotomy there. And then secondary is furthering the idea that metabolites can have an active role. And in our case, we've been working on these cholesterol metabolites and how they may have a role in controlling NK cells, and so it depends what cells NK cells are in contact with as to whether they are exerting their optimal functions in terms of cytotoxicity. That leads me just to thank all the people that have done the work, and we've a great lab and a lot of great people in the lab, both past and present, including the people I highlighted, Katie, who I showed you some work of hers, Deanna did the dendritic cell work, we have Chloe who did some of the NK cell work as well, and some of the early work was by Ray and Simon, some great collaborators as well, of course, including Claire Gardner here, who we do a lot of work with. This is us on our last summer outing. As you can see, Irish summers aren't so great. It doesn't matter anymore now, we're not allowed out of the house anyway, so our coats aren't getting any use, and of course our funding for supporting the work. And I'm happy to take any questions, and thank you everybody for listening.
Thanks a lot, David. This was very interesting, and I think the people liked it. We had 250 people online, and they all stayed, so that's good to see.
That's always a good sign.
I like the part on the DC T-cell interaction where they get restricted glucose in your dendritic cells. And there's also a lot of questions on this particular topic, so I think we should probably start focusing on that one. First question by Sandra van Vliet. She's actually PI in our group also. She asks whether the effect on glucose degradation on DC cytokines is a direct effect, or whether it's due to a loss of IL-10, which obviously can also be downstream of mTOR.
Yeah, so certainly in the more recent experiments in the DC1s, we don't see the same thing with mTOR. So this, which is interesting. So if you inhibit mTOR in our DC straight out of the spleen, you don't see the same effect as thick-weight glucose. So a lot of the early literature suggests that glucose and mTOR were similar, but a lot of that again was with sort of slightly less physiological dendritic cells. In terms of IL-10, we haven't looked at that directly. It is quite a short activation period, but we think it's probably linked to some sort of stress response. So we do know that when you take away glucose, you get upregulation of ER stress, and those have been linked to increased cytokine production in the past. So we think it's something to do with a stress response that seems to be beneficial for these cells. Mm-hmm. As far as I can remember from Diana's look at the lyses and the IL-10, I don't think IL-10 was hugely altered in when we looked at that. So I think it was using a cytokine beta array. So I don't think I think it is direct. I think it's through stress response, but I couldn't be sure.
Yeah. Another question related to this environment with low glucose. Francesco Colucci is asking, so you have these DCs that are obviously restricted with glucose, but in the end, the T cells that they activate will also be nutrient deprived. So what is the effect there of the starvation, and how do you see this?
Hi, Francesco. How are you? So first I should say that, so in those in vitro experiments, we're very clear that, we restrict glucose from the dendritic cells, then we wash them out, and when they see the T cells, there's loads of glucose. So in that situation, in a situation where we did the co-cultures and looked at the mTOR activation, in that situation there wasn't a global impact on glucose levels. So this is when we did the confocal imaging, and those lovely images. So glucose wasn't depleted in the whole media. What we're saying is this is really on a very acute microenvironment level. So for instance, so we don't think that the T cells are depleted of glucose. We think it, and that would make sense if you can think about a clustering happening around a dendritic cell. So there will be a preferential supply of glucose towards the T cells, but perhaps not to the dendritic cells. Now, I think it's going to be a lot more complicated than just a single nutrient when it comes to an inflammatory lymph node. I think you can imagine there may be that the dendritic cell may have low levels, maybe not zero levels of any nutrient, but it may have low levels of multiple amino acids and glucose that could lead to responses such as ER stress.
And personally, I was also wondering how you align this with Erica Pierce's work in Cell, where they show that the glucose competition within the T cells actually also dampens anti-tumor responses. So who's winning that battle, actually, you think?
So in the tumors, absolutely. So no, I think if you deplete a T cell of glucose, they don't work. Absolutely. But I think the concept here is that it's not like in a tumor. There's very little glucose anywhere. It's if you talk about an inflammatory lymph node, the idea is that you have very discrete gradients of nutrients, such that the T cell takes the glucose and the dendritic cell is without. So we don't think that there's a situation where in that context where T cells are deprived of glucose.
Yeah, exactly. So it's rather a location there.
Mm-hmm.
Thomas Rogers, he also comes with an interesting question. He says, in the context of an infection where you have a lot of neutrophils, which are very highly glycolytic and can maybe also deplete glucose in the microenvironment, might this actually contribute to enhancing DC stimulation of T cells?
I don't think so, but I will say one thing about neutrophils. I love neutrophils. I think they're a fantastic cell. And they're really interesting because neutrophils are absolutely packed full of glycogen. So there's some old electron micrographs of neutrophils, and they are packed full of glycogen. So my idea, and they are very glycolytic, but I would say that perhaps their glycolytic not by using external glucose, but by using the glycogen stores that they have. And they're short-lived, so, you know, I've no evidence of this, but I always like to think that neutrophils, they arrive at a site of infection, they start going about their business, and they use their internal fuel stores, all that glycogen, and once they run out, then they die and they undergo netosis or whatever it is. But I would say they're probably not a huge utilizer of external glucose, whereas an activated T cell is. So they have no, you know, a naive cell has no glycogen stores. You activate it in the first 18 hours, and then it goes massive blastogenesis, and it's sucking up huge amounts of glucose and other amino acids. So I don't think neutrophils are necessarily going to be contributing, but, you know, I don't work on neutrophils, but that's just from the analysis of all the literature.
Right. And then there's also a few questions on how this glucose and glutamine restriction would impact on utilization of fatty acids, but also ketone bodies, and how could that drive immune cell function? Did you see changes there? Did you look at that?
Yeah. So that's complicated. So the reality is, once you get in vivo, it gets so much more complicated, because you have all these additional stuff. So we haven't, you know, at the moment, it's really about trying to prove a core concept that, you know, nutrients can become limiting within these really, really sort of precise microenvironments. So yeah, absolutely. So there's an argument for looking at a whole host of things that could be competitively depleted around the dendritic cell. You know, T cells love methionine, they take up lots of serine, so all these things. So and, you know, and then there's also these other metabolites that could be looked at. So we haven't scratched the surface yet. But, you know, a real challenge is that microenvironment within the lymph node is very complex, and we just actually don't have the tools to know what goes up and what goes down at that sort of sort of single cell level. So, you know, we really need to, and that's, I think, is a limiting thing in immunometabolism at the moment, is to be able to know where the nutrients are, what cells have them, what cells don't have them. And at the moment now, we're really limited to taking the cells out, you know, sort of purifying them and sticking them into machines, and so or else, you know, I guess we can do metabolomics on a sort of a tissue basis, where you can, you know, there's some of this really fancy new mass spec techniques where you electrospray, you blast the tissue, but it's not single cell yet. So really, this concept of single cell competition for nutrients, it's really difficult to work on at the moment.
Yeah, that's something we'll see popping up probably in the next few years. There's some interesting things coming on there. So that's nice to see. You see this double metabolic shift, so an increase of both ECAR and OCR. And I think Jean Lima sees something similar when depleting a metabolic sensor like sirtuin. So the question there is, can I expect a more pro-inflammatory profile of this disease?
So so the DCs, there wasn't. So I didn't show any metabolism on the DCs. So the DC metabolism been done mostly by Bart Everett's group and Ed Pierce. So in that situation, there's, you know, I think it's, you know, most of that's been done on bone marrow DC. So I think we still need to know what exactly is happening in the metabolism of DC. But in the NK cell, absolutely. So there was a robust increase in both glycolysis and OXPHOS. And that's not surprising, you know. So often people talk about this glycolytic shift. In a lot of cells, there isn't a glycolytic shift, you know. There's not a Warburg effect. It's really an upregulation of both. If you look at active T cells, they do a lot of OXPHOS as well. So it's not the same as the Warburg effect, which was first seen in tumor cells, where, you know, there was no real observable OXPHOS. And, you know, there's very few cells, maybe the M1 macrophage, which is where they turn off OXPHOS and do glycolysis. That's a proper Warburg effect. But yeah, so in a lot of cells, you see a ramping up of both pathways, where it's really just they're ramping up all pathways to meet the metabolic needs, and for T cells in particular, that's about rapid growth and proliferation. In these NK cells, it's also about rapid growth and generating lots of biomolecules for their effector functions.
Good. I think we should somehow conclude. Maybe I have a last quite conceptual question. So during your introduction, you also mentioned this signal 1, 2, 3 of T cell activation by dendritic cells. So should we now consider metabolism as a signal 4, or maybe have a signal 0 without the proper metabolic pathway, no signal 1, 2, 3? How do you look at this?
Well, that's the exact spin I gave my ERC grant. So yes, I called, but I called nutrients signal 4. So okay. potentially, yeah, you know, levels of nutrients could be another signal that helps to shape the nature of the T cell response. But yeah, so, you know, but again, of course, until we can actually visualize nutrients in that lymph node, it's going to be very hard to see that whether that really happens in vivo. And we're, you know, that's something we really want to find out in the next couple of years. But yeah, I would agree with that. I like that that idea. Yeah.
Okay, nice to hear. I think we will conclude here. Thanks a lot, David. I really enjoyed, and based on the questions I see here, I think people really like your talk. There's quite a lot. I will share them with you, so maybe you want to follow up on a few. For the audience, please visit the website to be updated on the program. Next week we'll have Giuseppe Matarese. He will be talking about regulatory T cells and metabolism. And also for May, we're already quite fully booked. The only free spot we still have is the 13th, and that's actually the day we planned the physical meeting. So we're now considering to maybe give PhDs and postdocs a chance to present in 15 minutes or so. So if there is interest in this, let us know, and we might consider to take that spot for junior researchers. And for the rest, yeah, we're happy to get feedback from you. And if you have suggestions for people to invite, also let us know. So that was it for this week. I really enjoyed. I hope you did too. And I look forward to see you all again next week. So have a good week. Have a good day. Bye all.
Take care.